Welcome back for more AP Biology. We're going to deal with how cells talk today. In particular, how come one set of information, so one signal, can have multiple responses. It's something that the College Board really likes to test. So, how do we know that cells talk? I presented previous bits that suggest that there's communication, but let me give you a totally different twist. What you see on the screen is something referred to as a slime mold. They're not particularly big, but they're made out of single cells, and they're single-celled organisms. But every once in a while, they start to aggregate together, and when they aggregate together, they make large structures that look like that. That's actually not a big piece. It's not a whole bunch of slime mold, so it looks like it's like an overwhelming mass. It really isn't. But somehow, these things go from being individual cells to aggregating together to function like it's one massive organism. Well, how? The only way to explain that is they're talking to each other somehow. So when you communicate, you need to have three things go on. First thing is you need to have some type of signal, you need to have some type of message to give. You're going to need to have some way of processing that signal, and then you react or you need to respond to the signal. If you can have those three things happen, communication is possible. If one of those three things fails, there's no such thing as communication. So when we look at cells, what do they have? Those three components. This is typically what we call cell signaling or signal transduction or cell communication. There are three components. The first one is what we call the reception, which is when the signal, whatever the signal is, binds to something that says, hey, there's a signal. We call that a receptor. The second component is what we call the transduction or an amplification pathway. That's where you process what's going on. And then the third thing is you respond to it. The response can either be doing something with your genetics or increasing the amount of chromatin you have, or we start releasing vesicles, or we start making vesicles, or the cell changes its shape, or the cell divides. That all those things turn out to be responses. So how does this turn out to work? So if I were to take a cell and add a molecule to it, a signal molecule will bind to the receptor, and the result is we see a series of responses or reactions going on inside the cell that ultimately result in some type of cellular response. Something is going to happen. But if I were to take that same signal at a totally different cell, what I could see is if the receptor is different, so it's same signal, different cell, different receptor, if it binds to that different receptor, what I get is a totally different set of pathways that get triggered. The thought process is different. It's much like in class or on this video, I can be talking at you. I'm attempting to communicate. Some of you are processing and saying, oh, I understand exactly what he's saying. So you would sit there and you'd nod, or you'd say, yeah, I understand what you're talking about. Others of you are taking it, you're attempting to process it, and just, nope, this doesn't make any sense at all, either because I'm wrong or because you're completely lost. But we're having two individuals receiving the same signal, and we have two different outcomes. The difference is how you process it. So different cells can process the same signal a different way. How do we do that? A different receptor. So if we really look at this is a real example of cell signaling pathways. So this one's going to deal with cancer, and you look at it and you go, I don't think I like this anymore. So when we look at these, you see all these lines. What those lines deal with are proteins turning on other proteins. They're activating the proteins. What we're actually seeing is the transduction side, the processing. And you'll notice that there are some arrows that point around, and they end up resulting in little P's being added. Those are phosphates. So when we add phosphates, for the most part, it's not entirely always true, but for the most part, what you could do is add a phosphate, and that ends up turning on a protein. In some cases, it turns it off, but in a whole bunch of cases, it turns them on. The ones that are where you add the phosphate and then it becomes activated, we call those kinases. Much like in the previous lecture. And if you look through here, you actually will see some things that you might recognize, such as cyclins. There are cyclins in here because this is dealing with cell replication. All of this is showing us that the amplification side is complicated. It's very complicated because there's lots of parts involved. The signaling side, pretty simple. We can put the signals into basically two categories, chemical signals. One of them we call steroids, one of them we call proteins. The steroids are fatty substances. They're going to be things like testosterone and estrogen and cholesterol and things we can make from those. Where do they bind to their receptors? Where are their receptors? If you were to go hunting along the cell membrane, you wouldn't find them because steroids are fatty substances and fatty substances can dissolve through the cell membrane. So where do we find these steroid hormone receptors or these steroid receptors? Inside of the cell because they can reach the inside of the cell. Protein signals or things protein-like, so polar compounds or large charge compounds, they don't get the option of making it through the cell membrane because the cell membrane is a barrier to those things. The result is they need to have their receptors on the surface of the cell. We will have slightly different amplification pathways because the signals are being received in different locations, but we could potentially get similar results from a protein signal and a steroid signal. So that's the basis of, or the basics of how cells talk to each other. You add a signal, binds to a receptor, we get an amplification, a transduction pathway, you get a response. That seems pretty simple. Well, what are the different ways that they could talk to each other? They could either have direct contact, that could be through a cell-cell, or this strange version referred to as a neuronal messaging, which is when neurons talk because they have a very unique way of talking to each other. We could have things that we call paracrine, which are cells somewhat near each other, or we could have long-distance communication. Those are referred to as endocrine and exocrine. Here it doesn't show exocrine. Endocrine is when we dump the signals into tubes. Exocrine is when it's not dumped into a tube, meaning it's out in the open. Once upon a time ago, we talked about pheromones, and pheromones turn out to be a signaling compound that is useful in cell communication.